The genetic blueprint of life is made up of 4 letters, and just these 4 letters alone can form almost an infinite number of combinations in our genome. This is the transcription process where DNA gets turned into protein. RNA polymerase finds corresponding RNA base pairs to a DNA sequence, making messenger RNA. This mRNA leaves the nucleus through the nuclear envelope, where it then finds a ribosome. The ribosome brings in corresponding tRNA molecules with amino acids, which then form a sequence, and this sequence of amino acids undergo a series of conformational changes to form a protein. This process is important because it could dictate the difference between a mutated cell versus a normal cell based on which proteins are expressed and whether these proteins are misshaped or not. And so this is caused by differences in gene level expression. And to study this, we would use something called high throughput sequencing, specifically with RNA sequencing, to see which genes are active or transcribed and how much of these genes are transcribed. So to complete high throughput sequencing, there are 3 steps. First, prepare your sequencing library, so gather the data from a real life organism. And then number 2, sequence and then normalize the data. And step 3, perform data analysis. So I didn't have an actual RNA sequencing machine, so I just did step number 3 based on a pre-made data set of fruit flies. So to do this, there are 6 steps. Number 1, get your RNA from your cell. Number 2, break these RNA into smaller fragments. Number 3, convert each of these small fragments of RNA into double-stranded DNA because DNA is more stable and also easier to modify than RNA. And then you would add sequencing adapters to each of these small fragments on both sides. And the reason why you would do this is because it allows your RNA sequencing machine to recognize what each of the fragments are and that they need to sequence it. And then it also allows you to sequence multiple samples at the same time because you can add different sequencing adapters for different samples. Then step 5, use PCR polymerase chain reaction to amplify the sample. And then step 6 is quality control. So you have to verify that the concentration and also the length of all the samples are relatively similar. Step 2, once we have our data, the sequencing library, we will put it in a sequencing machine, and all of these individual fragments, usually over 1 million, are arranged vertically in a cell called, or a grid called a flow cell. And what the sequencing machine does is that it has fluorescent probes that are color-coded based on which type of base out of the 4 that it binds to. And so you can imagine that you would have a vertical sequence and then it would bind these fluorescent probes from bottom up, and then each time it binds, the machine would take a picture. So by the end you have a series of colors, and we would convert those colors into their corresponding base pairs to determine the sequence of each of these individual fragments. So now we have our data, but the problem is there are often things called garbage reads, basically data that isn't useful or has been skewed in some way. And an example of this would be that the sequencing adapters, instead of binding to the DNA fragment, bound to itself. So we would be sequencing useless information. Obviously, we would need to take that out of our data set. And then we do something called counts per read, which basically means that we take our sequencing data and then we map it to a genome of, let's say, a plant or an animal or a human, and then you would determine the location and name of each gene based on its sequence on the type of chromosome and the location on the chromosome based on the genome. And so you can imagine that you would have an array of data, one column of all the gene names that you sequence, and then a bunch of other columns of all the samples that you obtained. So here is why we need to normalize the data after this step, is because, for example, for sample 1 in this column, you would have more samples of that specific gene because they were higher quality reads, and then in sample 2, you would have less high quality reads, so less samples of that specific gene. So we need to equalize this in some way, and the easiest way to do this is to divide out the number of samples you got by the total number of samples. But in real life, there would be other forms of calculations to do this. To perform the data analysis without using a command line, I used an online software called Galaxy. And so what I did was first I uploaded the data onto the platform. And then the data set basically included the sequence data of chromosome 4 of the fruit fly, and then also uploaded an annotation file, so basically the entire chromosome 4 of the fruit fly sequenced. And then after this, I took each of these individual RNA sequenced data sets, and then I performed alignment with a tool called HiSet2. And what this does is it takes all of these individual fragments and aligns them basically if to say they would be part of the same mRNA fragment, but some of them are introns and some of them are exons. And it would all help me locate all the splice sites, and I used a tool called JBrowse to visualize the alignment of each of these RNA fragments. And so next step would be quantification, where we take each of our RNA sequences and then we map them onto the reference genome. And specifically in this case, it was chromosome 4 of the fruit fly. And this allows us to determine key chromosomal features of each gene. For example, whether it's a protein coding gene or a pseudogene, or if it's an intron or an exon, and then where exactly on the chromosome it starts in terms of base pairs, and then where it ends. And then based on this data, we would generate a count matrix, and then that count matrix, if uploaded onto another piece of software, it will allow us to create different plots such as heat maps or MA plots that will allow us to visualize the data. So I used an online software platform called Zgust, and it allowed me to take my count matrix and then map it onto various forms of visualizations. So one of them is an MA plot, and basically if you look along the x-axis there are blue dots and also red dots, but basically the closer together the dots are, it indicates that the closer or more like the genes are to each other, and then the further along the y-axis, for example the red dots they are, it means that the genes are more different from each other. And this can also be visualized on the heat map below, and you can see the color gradient. The color intensity represents the level of difference between the gene expression of the different samples of the same genes from chromosome 4 of the fruit fly. So that might have seemed like a relatively useless process because it was just for fruit flies, but if we repeated the same process with the human genome, we can analyze differences in allele expression or gene expression, and it will allow us to extract an incredible level of insight because what we're looking at is the transcriptome, all the different sequences of mRNA in the cell, rather than just the genome or the DNA. So this level of insight will potentially lead to different cures, novel cures for genetic diseases, or even different treatments for cancer based on gene expression levels. So really, it holds a lot of potential. And that's all for today. I hope you learned something new and you enjoy the video. See you next time.